Good morning, everyone, and for you, I guess it's good afternoon. It's about to be 5:30 over here in Canada. I'm Sabina Halappanavar. I'm research scientist from Health Canada in Ottawa, in Canada. And it's nice to be with you all, and know that you all are doing well and being safe. And thanks for the opportunity to virtually present it. It's too bad I can't see you all and you can't see me, but we'll do with that. So, as Ulla already mentioned in her presentation, my topic for the discussion is going to be the AOPs and the key events for in vitro endpoint design and development. I had a discussion with Barbara, and because she's going to be focusing specifically on lung fibrosis and some of the in vitro models that she has developed. I will focus on something at approach that we have taken to develop a predictive model for lung fibrosis.
Sorry, Sabina, can you keep your mic closer? It's a bit
okay.
Sound is a bit unstable. Yeah.
Can you hear me now?
Yes, it's better. Yeah. Thanks.
Okay. So as you lack of concordance in what's assessed in vivo and in vitro. So the figures over here show what's most assessed in vivo versus in vitro. So the size of the letters is directly proportional to the number of times the particular endpoint is assessed. And the figure itself is assembled from approximately 2,000 papers. So as you can see that in vivo, organ toxicity is mainly targeted by endpoints such as body weight, histopathology, which are basically the endpoints of relevance to regulatory decision making. But whereas in vitro studies, you will see that the endpoints are basically targeting the biological processes or the basic functions of the cells, such as cell death or the oxidative stress or cytokine induction, etc. And as you all know, in vitro studies are conducted mainly with the purpose of understanding the mechanisms of action, and therefore not always have relevance to the in vivo observations that we see in vivo studies, as well as use for the regulatory decision making. So how do we go about bridging the gap between the in vitro and in vivo, or how do we go about making in vitro endpoints relevant for in vivo responses? One of the ways is by using AOPs framework. So for example, Ulla already mentioned that AOPs it's a conceptual framework that allows you to systematically organize the existing mechanistic information in a linear and modular format consisting of several key events that are causally related to one and in each other, and they show you the path from the exposure to the eventual adverse outcome. So and they will help you, and because of that mechanistic richness of the AOP, they are able to help you to anchor the in vitro endpoints or in vitro observations to the in vivo responses or the observation. So well-developed AOPs, and I think this was already discussed by Ulla briefly, that well-developed and well-established AOPs have far-reaching applications in the human health risk assessment of nanomaterials. So when you have a quantitative AOP, and when there is sufficient exposure, material characterization, and dose response information, the well-established quantitative AOPs can be used to derive risk indicators important for the risk assessment process. But the putative or the qualitative AOPs, which may not be quantitative per se, but also can be very informative and can be used to identify data gaps, prioritize research focus, for example, or the data from the putative AOPs can be used as weight of evidence to support read across categorization decisions. And more importantly, putative or semi-quantitative AOPs can be used to help support the design and development of targeted in vitro endpoints, as mentioned by Ulla in her talk. So as you saw in Ulla's presentation, there were 5 AOPs that were developed by SmartNanoTox along with Health Canada, and they are they capture the 4 adverse outcomes of relevance to inhalation toxicity of nanomaterials. These AOPs form networks, form a network connected by commonly occurring key events across the AOPs, really overrepresented KE nodes across these AOPs. As you see here, KEs of inflammation, the molecular initiating events, KE of oxidative stress, as well as KE of a tissue injury form the KE nodes. So here you have 2 choices. One, you use the individual KEs in the linear AOPs that I showed you in the previous slide to design targeted in vitro endpoints and assays, or use the information from the network of AOPs as shown here and focus on the KE nodes from the network for designing of the targeted in vitro endpoints or assays. So in vitro endpoints targeting the KE nodes from the network can form the first-tier screening of nanomaterials, and in vitro endpoints targeting the downstream KEs in an AO-specific manner can make up the second tier, confirming the material potency to induce specific adverse outcome. So in fact, KE nodes can also be targeted for in vivo, for in silico QSAR modeling. For example, later on you may see in Thomas's presentation that even though MIEs are targeted for QSAR modeling, one can also choose KEs, and in this case, a KE from the KE node that is overrepresented across these 5 AOPs may be very useful for targeting by the QSAR model. So at Health Canada, however, we used a different approach to come to the stage of developing predictive in vitro models. As many of you, oh, sorry, I'm a little bit ahead of myself. In this slide, what I would like to bring your attention to is that we have on your left, you will see the overlapping case from the network. In the middle, we have assays that are available, in vivo assays that are available at present to assess these key events, suggesting that some of these key events are assessed and targeted by the in vivo studies, and there is test guideline, OECD test guideline available for these for assessing these endpoints. And also specific assays have been mentioned in the test guidelines. And on your right side, you will see various in vitro endpoints which are not validated, for which there is no test guideline available, but nevertheless, the many endpoints and assays and models and systems are available in the literature that can be used to assess these key events that are from the network of the AOPs. However, like I was saying, that at Health Canada, we have used a slightly different approach to identify the endpoints of interest and develop a predictive model that would help us predict lung disease after exposure to nanomaterials. So we basically used high content transcriptomics and bioinformatics to develop AOPs, and from there we use the AOPs and the specific key events identified in the AOPs to establish a gene panel that can be used in vitro to assess the multiple KEs in the AOP. So as many of you are aware that we at Health Canada have the largest in vivo lung transcriptomics database for nanomaterials. And this is all thanks to Ulla and Håkan's long-standing collaboration from NRCWE in Denmark, actually. So we practically have lung transcriptomics data for more than about 75 individual nanomaterials using the mouse model. And we use these transcriptomic profiles of lungs exposed to nanomaterials and combined it with transcriptomics profiles of lungs exposed to lung disease causing substances and to reveal the identity of nanomaterial-induced lung toxicity. So you can see that in this particular figure, the circular hierarchical clustering shows that some of the responses induced by nanomaterials resembled like bacterial infection or bleomycin injury early after exposure, and later exposure to some of the nanomaterials resembled those of Th2 response. And you all know that the bleomycin injury or bacterial infection or even the Th2 responses, they all lead to lung fibrosis. So combined with information from the literature and the mechanistic information that we derived from we derived from this meta-analysis of the transcriptomics data, we established an AOP, AOP173 for lung fibrosis. And I won't go into the details of the mechanism itself, as Ulla has already talked about it. But from here, in parallel, we expanded the transcriptomics meta-analysis database and included not only the diseases such as lung fibrosis or asthma, but we also included other diseases such as lung cancer and tumor formation. And we had about 75 different publicly available studies, and we conducted another kind of a statistical analysis that we call bi-clustering analysis, from where we derived we identified clusters of gene signatures that are specific to certain diseases, including cancer, fibrosis, inflammation, asthma, etc. So from this bi-clustering analysis, we came up with a 17-gene panel that we think and we show is predictive of lung fibrosis as we see it in the, as we have explained it in the AOP173. So the 17 gene panel, of course, has consists of 17 individual genes that are associated with the multiple key events in the AOP173, and therefore is much suitable for assessing or predicting lung fibrosis using in vitro or ex vivo models. So many details of the process of establishing this lung fibrosis predictive signature, as well as the details of the genes, are described in a recently published paper in SMALL, which is an open access publication, so you can read it. And there are about 34 supplementary files showing you the various different ways and different studies that we have used to validate this particular signature. Just to briefly describe what you see in this heat, what this heat map shows is that the green and the red are the individual genes as shown on your right side. There are 17 genes there, and at the bottom are the studies that are used in establishing the training set to establish the predictive gene panel. And on top you have two rows. At the bottom it's a class prediction, that is class assignment, that is we have assigned each of these studies a class which is either non-fibrotic or fibrotic based on the information from that particular study. From that particular study. And on the top, it's the prediction class that is assigned by the predictive signature, the 17 gene panel signature. So using the 17 gene panel, we predicted whether or not a particular study is indeed non-fibrotic or fibrotic. And as you can see, the 17 gene panel accurately predicts the studies that are pro-fibrotic, except for one of them. which was assigned by us the profibrotic class. So in a sense, it says that the 17 gene panel is well suited to predict the lung fibrosis using the in vivo or in vitro models. So then we validated the PPS17, the 17 gene panel, using bleomycin studies. Again, the heat map shows the genes on the left on the right. At the bottom, the types of studies used. And on the top, you have the class assignment as well as the prediction assignment by the PPS17. Actually, the bleomycin studies are on your rightmost side, and you can see that there are many bleomycin studies are positively identified as fibrotic, and some of them are non-fibrotic. And non-fibrotic studies were mainly belonging to the experimental groups of low doses or early exposure, post-exposure time points when the fibrosis was not yet established, I think. We also validated the PPS17 using nanomaterial studies, basically the multi-wall carbon nanotube studies. And I think we had about 82 or 84 conditions in there. And as you can see, there are many reds and many more blues, suggesting that we could identify few of the CNT conditions as profibrotic, but many number of them were non-fibrotic. Again, non-fibrotic conditions were mainly from the low dose exposures and the early time point, early post-exposure time points. So with this, we could, even though because of the limited number of studies available, transcriptomic studies available, validation can never be that effective. However, we can say that we were able to validate the PPS17 using as many number of studies as possibly available from the public domain. So then, and although we didn't include it in our study design, because of the courtesy of Tobias, we were able to understand where identify the cell types in the lungs which express these particular genes. Tobias actually conducted an analysis using one of the using the single cell sequencing data that he has from their own studies, where they have lung cells exposed to bleomycin, and he ran this 17 gene panel in those cells and in that particular model, and showed that 17 genes are expressed, all 17 genes are expressed in bleomycin exposed lung cells, and they are exposed, they are expressed at varying degrees. As you can see here, 3 of the genes that I'm trying to show you, ARG1CT and REC1A, are all expressed at high levels, but at varying degrees. And some of them are expressed in multiple cell types, which are reflected by the circles on the sidebars here. And the size of the circle reflects the amount of expression in that particular cell. And some of them are highly expressed, and some of them are lowly expressed, and some cells are highly expressed and some of them low. So what it tells me is that the lung fibrosis as a disease is brought about by the interaction of multiple cell types in lungs, and therefore may not be absolutely captured by using a single or a co-culturing of a couple of cell types. And it may require actually an organoid culture, which is what we used at Health Canada to as an ex vivo alternative to in vivo assessment of the lung fibrosis induced by nanomaterials. So we isolated the lungs from the mouse, and we sliced them at the 250 or 500 micrometer thickness and cultured them ex vivo and expose them ex vivo to different types of nanomaterials. And because ex vivo lung slice consists of all these cell types in a lung, from a lung, it can act as a lung mimic or mini lung organ, and it has the normal architecture, original architecture of the lung with all the cells in their anatomical positioning, and therefore is much more suitable for assessment ex vivo. Although it does not completely eliminate the use of animals, it does reduce substantially the number of animals required for the assessment of toxicity. So we exposed the lung slices to multi-wall carbon nanotubes, and you see that in the microscopic image here, carbon fibers interacting with the lung slice, just the way you would have seen it in vivo, where fibers are piercing through the lung slices. This indicates that the molecular initiating event, which is described in AOP173 as the interaction of materials with the lung microenvironment or lung cells, is satisfied. Then we also looked at induction or expression of several pro-inflammatory mediators using bioplex assays, and we show that this little heat map shows you that several pro-inflammatory mediators are expressed following exposure to Mitsui-7 as early as day 1 post-exposure, and continue to be expressed after day 7 post-exposure, and which are also seen or induced by in a bleomycin-exposed lung slices, showing that KE1 and KE2 that describe the inflammatory events in the AOP173 for lung fibrosis are induced in the lung slice. Next, we looked at the induction of PPS17, which is the 17 gene panel, and in the Mitsui-7 as well as bleomycin exposed lung slices. Here you can see that several of the 17 genes are well expressed following exposure to bleomycin, and their expression goes higher and higher with the days, with the post-exposure days. On day 7, you see a lot of redness, which indicates high expression of some of these genes in bleomycin model. In comparison, 7 itself induces fewer of these genes, and only the expression of these genes becomes apparent only on day 7 and less so during the earlier days. However, this suggests that the 17 gene panel that targets the multiple key events in the AOP173 can be triggered and assessed using the PCLS, warranted that further studies have to be conducted to optimize the multiple carbon nanotube interaction with the cells. Since these PCLS slices, the lung slices, are exposed in submerged conditions, we may have to work around those conditions to make sure that there is optimal interaction between the nanomaterials and the lung slices. Finally, we were able to show that the lung slices show the manifestation of fibrosis following exposure to Mitsui-7 and bleomycin by day 7. And this is shown on the upper panel of the histopathology panel by the Masson trichrome stain. And at the bottom, you can see the localization of macrophages, activation of macrophages in the various areas of the lung slices. So histological evidence is one of the important evidence and gold standard used by the regulators for the risk assessment. And by having this ability to induce the disease and observe the manifested disease by the histopathological observation makes the PCLS an important predictive model for assessing the lung fibrosis. And by satisfying all the key events that are explained in the AOP173 PCLS model actually aligns with the AOP framework, and therefore may be used for the important decision making in the interim for read across or for prioritization or even categorization of nanomaterial in potency. So with this, I would like to thank various members of my lab who have worked over the years to bring us this far. Health Canada funding, which is from Genomics Research and Development Initiative, as well as Chemicals Management Plan. And more importantly, I would like to thank the SmartNanoTox Consortia for the wonderful opportunity of collaboration that they provided me, for welcoming me in their consortium and allowing me to contribute to your objectives. The big thanks to coordinator of the SmartNanoTox, Vladimir, for this wonderful collaboration that we have shared over the 4 years. Also thanks to Ulla Fogel. She has been my partner for many years now, and many AOP developers who allowed me to use their AOP in the review that we recently wrote, as well as in this presentation. And also for Tobias, who has generously helped analyze the data using his own single cell genomics data and helped me to identify the cell types that express the 17 genes in the gene panel. So again, thank you all for being there virtually and listening to the presentation.